Hi everybody. Let us continue with the chapter Sexual Reproduction in Flowering Plants. So in the previous video we stopped till the process of double fertilization. So let's recollect what is double fertilization. It is nothing but the combination of syngamy and triple fusion. You remember what is syngamy and triple fusion? Both the events happen inside the embryo sac. So first let's talk about the triple fusion. Triple fusion is nothing but the fusion of one of the male gamete with the diploid central cell, the polar nuclei in the central cell. So the polar nuclei is a combination of two nuclei. So it is diploid in number, and this fusion results in the formation of primary endosperm nucleus PEN, which is triploid in number, and this is found inside the primary endosperm cell. And this primary endosperm cell in turn develops into endosperm, which acts as one of the nourishment tissue or nutritive tissue for the developing embryo. On the basis of the development of endosperm, it can be classified into three types: nuclear endosperm, cellular endosperm, and helobial endosperm. Let's check this one by one. In nuclear endosperm, the primary endosperm nuclei will keep dividing, and after the division of the nucleus, there is no cell wall formation. So cell wall formation is not followed by the nuclear division. And as a result, nuclei remain free in the cytoplasm, and hence they are called as free nuclear endosperm. And this free nuclear endosperm is usually found in coconut. Inside the coconut, the watery form, that liquid form inside the coconut is nothing but the watery liquid endosperm. And this liquid endosperm is nothing but the free nuclear endosperm. And you know what the kernel is, the white hard kernel? It is nothing but the cellular endosperm. So what is cellular endosperm? In cellular endosperm, the cell wall formation occurs after each division. So you can observe in this figure that after each division, the cell wall formation is formed, and it has got the shape of a complete cell. It has taken the form of a complete cell. So this kind of endosperm is called cellular endosperm. So the kernel inside the coconut is an example of cellular endosperm. The next kind of endosperm is helobial endosperm. So this type of endosperm is found to be intermediate between the nuclear and the cellular type. And in this endosperm, helobial endosperm, the first division of the primary endosperm nuclei is is accompanied by the cell wall formation. So it divides into two, followed by the cell wall formation. So it gets differentiated into two cells. And this division is actually unequal. So it divides the embryo sac. Imagine this is the embryo sac, and this divides the embryo sac into small chalazal end and bigger one. When compared to the chalazal one, the micropylar end is found to be a bigger one. And free nuclear division is followed in both the chambers, that is micropylar, the chalazal as well. But comparatively, the divisions inside the chalazal chamber is less than the micropylar chamber. And later, the free nuclei inside the chalazal end seems to disintegrate. At the same time, the micropylar end, the nuclei formed, will develop cell wall. So it gets converted, or it transforms into cellular endosperm. Thus the cellular endosperm tissue is formed near the micropylar end. So this is how helobial endosperm is formed. Now let's talk about the embryo formation. So how is embryo formed? Embryo is formed as a result of syngamy. So syngamy is nothing but the fusion of one of the male gamete and the female gamete, that is the egg cell, and this results in the formation of zygote, which in turn develops into embryo. Now let's talk how the dicot embryo is formed. So there is difference in the stages of development of dicot and monocot embryo. So first let's talk about the development of dicot embryo. Firstly, the zygote divides transversely into two cells. So imagine this is a zygote which is diploid in number, which divides transversely into two cells. Consider this as the apical cell and this one as the basal cell. And this basal cell, this end is supposed to be the micropylar end, and here it is the chalazal end. And now the next stage, the basal cell keeps dividing transversely, whereas the apical cell divides longitudinally. There is a longitudinal cell formation, cell wall formation in the apical cell. So this basal cell continues dividing and it forms a suspensor, where the apical cell, which is at the chalazal end, keeps dividing longitudinally at its 90 degrees of each cell and then forms a globular embryonic stage. It results in the formation of globular embryo. It is actually pro-embryo. It's not completely developed. And this globular pro-embryo keeps dividing, whereas the suspensor formed from the basal cell helps the embryo gets attached to the embryo sac, and the globular region keeps dividing and it forms the heart-shaped embryonal stage, where this region keeps dividing. Now it develops into the mature embryo, or it gets matured with a suspensor and then the embryonal axis and two cotyledons with all cells inside. So this is a mature embryo, mature dicot embryo. This region is the embryonal axis, and these both are the cotyledons. So since they have two cotyledons, it is recognized as dicot embryo. The part of the embryonal axis above the level of the cotyledon is the epicotyle, which terminates with the plumule. So epicotyle here terminates with the plumule, whereas the cylindrical portion below the cotyledon is hypocotyle, which terminates at its lower end in the radicle. So this portion is radicle. This is the figure given in your textbook. The zygote is dividing into two. Suspensor is formed, heart-shaped embryo. The suspensor is there in the top, and heart-shaped embryo is developing into the mature embryo with the radicle and plumule and with two cotyledons. So this is how the dicot embryo is formed. Now let's talk about monocot embryo. A typical monocot embryo has only one cotyledon. So the cotyledon in monocot, we call it as scutellum, and it is situated towards the lateral side of the embryonal axis. So this is the embryonal axis, and scutellum, which is the cotyledon, is found towards the lateral side of it. And in the embryonal axis towards the lower end is the radicle with a root cap, and an undifferentiated sheath is found, which is called as scoliorhiza, and the embryonal axis above the level of the attachment of scutellum. So here the scutellum is attached. Above the level of the attachment of scutellum is the epicotyle or the coleoptile, and this has the shoot apex, that is the radicle. And so the sheath found here, it is a leaf primodia or the primary leaf, and it is coleoptile. It covers the shoot apex. And you can see an epiblast here. The epiblast is nothing but it is considered as a rudimentary cotyledon, that is underdeveloped cotyledon. So the well-developed cotyledon here, it is scutellum. So we consider monocot has only one cotyledon in it. So in angiosperms, that is in the flowering plants, after fertilization, the zygote developed into embryo, and this embryo is enclosed inside the ovule. The ovule develops into seeds. Seeds are formed from the ovules. According to the presence or the number of cotyledons, the seeds are classified as monocotyledonous seeds with only one cotyledon, and the other one is dicotyledonous seeds. That is, the embryo with single cotyledon forms the monocot seeds, and the embryo with two cotyledons forms the dicotyledon seeds. We can also classify seeds based on the presence or absence of endosperm in it as albuminous or endospermic seeds, ex-albuminous or non-albuminous or non-endospermic seeds. As the name suggests, in albuminous seeds, the cotyledons are very thin because the endosperm persists there and it nourishes the seedling during the early stages of development. Whereas in ex-albuminous or non-endospermic seeds, the food is accumulated in the endosperm tissue at an early stage, but later during the development of embryo, it is completely utilized by the embryo. So the seed remains without endosperm because the endosperm is completely utilized. In this kind of seeds, that is non-endospermic seeds, the cotyledons store food and it becomes thick and fleshy. Whereas in the albuminous one, the cotyledons remain thin because endosperm is rich in food and here the cotyledon is providing food. Mostly the monocot seeds are albuminous due to the presence of persisting endosperm. In the case of dicots, both albuminous and ex-albuminous seeds are found in dicots. Examples of albuminous seeds are the monocotyledon seeds like wheat, maize, barley, castor, etc. Whereas the examples of non-albuminous seeds are pea, groundnut, etc. Occasionally, sometimes in some seeds such as black pepper and beet, remains of nucellus is persistent. Nucellus is the tissue found inside the ovule before the development of the embryo sac. Before the development of embryo sac, ovule is filled with a mass of cell called nucellus. In some plants like pepper, beet, this nucellus remains or it persists and it acts as the nourishment or nutritive tissue. So this remains of nucellus is termed as perisperm. Now let's look at the structure of a seed. The integuments of the ovule, you know that seed is developed from the ovule. Ovule matures into a seed. So the integuments of the ovule, it hardens to form the seed coat, protective seed coat. And this micropyle is this region, it remains as a small pore in the seed coat. So through this micropylar region, the oxygen and water enter into the seed during its germination. Micropyle remains as a small pore in the seed coat, in a region of the seed coat, and through that micropyle, oxygen and water enter into the seed during germination. Now, if the germinating conditions or the favorable conditions are not available, the water is not available or any other conditions are not available, the seed will not germinate immediately. And its general metabolic activities slows down and then the embryo will enter into a state of inactivity called dormancy. It is identified that there are certain seeds which remain dormant and alive for hundreds of years. An example of that is lupin, that is Lupinus articus, a plant. The seeds of this plant is been excavated from the Arctic regions and it is recorded that after 10,000 years of dormancy, the seeds germinated and flowered. Similarly, another species of date palm, Phoenix dactylifera, or common date palm, recorded that a 2,000 years old viable seed been discovered and it germinated later. Once the favorable conditions arise, that is proper water, sunlight and oxygen is available, the seed starts germinating. Seeds break their dormancy and they start germinating. While the seeds are getting matured inside, the ovary also starts developing into fruit. So this is a typical fruit with a proper pericarp. Pericarp is the outer region of a fruit. We all know that ovary develops into fruit. So the wall of the ovary gets differentiated into pericarp. The pericarp may have an epicarp, mesocarp, and an endocarp. So this kind of differentiation of pericarp is seen in fleshy fruits like guava, mango, orange, etc. Whereas in dry fruits, the pericarp occurs as a thin sheath or thin layer or a scaly layer. The dry fruits like groundnut, mustard, etc. Do you think, is there any relationship between the number of ovules in an ovary and the number of seeds present in a fruit? Yes, there is. As the number of ovules, so the number of seeds, because seeds are developed from the ovule. As the number of ovary, so the number of fruits, because ovary develops into fruits. So there is a relationship between the number of ovules in an ovary and the number of seeds present in a fruit. We mentioned earlier that ovary develops into fruit and ovule into seeds. But there are certain cases where ovary does not develop into fruit. Some other parts would be developing into fruit. In this fruit, it's a typical fruit where the ovary develops into fruit and we mention it as true fruits. In certain other cases, like apple, strawberry, cashew, etc., the thalamus or the receptacle contributes to fruit formation. So such fruits where the fruits are formed from any other region or the fleshy portion is formed from any other region of a flower, then we mention it as false fruit. So in this case, this is a false fruit of apple. The region which we eat, this fleshy portion is actually developed from the thalamus and the real pericarp is found inside. So this is a false fruit of apple. What we eat is the false fruit of apple and the real fruit that develops from the ovary is at the center. And in the case of strawberry, actually it is developed from an inflorescence. It's an aggregate fruit. So all small or minute fruits which you see here are the collection of small berries or collection of small fruits. And this fleshy portion is actually the receptacle or the thalamus of the inflorescence. And you can see it is mentioned as achene. Achene is nothing but this fruits which cannot disperse on its own. So you can see a minute brown colored or dark colored seed surrounded by a small coating, and that is the actual fruit of strawberry, and what we eat is whole inflorescence. Such fruits which are developed from the parts other than ovary, we mention them as false fruits. And we know the fruits, seeds are developed as a result of fertilization. But do you wonder that certain fruits are developed without fertilization? So if fertilization does not occur, no seeds will be formed. Such seedless fruits that is formed without fertilization are called parthenocarpic fruits, and that phenomena is known as parthenocarpy. Parthenocarpy can occur naturally, and these days parthenocarpy is induced as well, so that we get seedless fruit, seedless apples, seedless grapes, seedless oranges we get as a result of this parthenocarpy. And this can be induced with the help of hormones and make the fruit seedless, which we all love to eat. But we know seed formation is very, very important, and it is advantageous for the angiosperms, right? Without seeds, no propagation may happen. Nature has evolved another special mechanism to produce seeds without fertilization. This method of production of seeds without fertilization is termed as apomixis. So here it's actually a substitution of sexual process by a sexual method. Here in this figure, the sexual reproduction is depicted, whereas the mother cell, which is diploid in number, undergo meiosis and it forms the egg cell, haploid egg cell, and it fuses with the sperm cell or the male, and it fuses with the male gamete to form the embryo, which develop into a new individual which is diploid in number. In certain cases, in some of the plants like grasses, variety of grasses, and some varieties of sunflower family, Asteraceae family, apomixis happen when the megaspore mother cell fails to undergo meiosis, or it does not undergo the reduction division. It remains as a diploid cell, and without fertilization this diploid cell will develop into a new individual, and it can be considered as a maternal clone because it has the same characteristics of the clone. So this significance of apomixis is used in the hybridization techniques where the desired quality has to be conserved for generations after. So in this, if a new desired trait is developed, that mother plant with the desired trait undergoes or induced to undergo synthetic apomixis so that the same clone shall be generated. In that case, no change in the desired trait shall happen, and for sure the clone produced will have the same desired qualities. And because of this importance of apomixis, it is widely used in seed industry, and active research is going on many laboratories to understand the genetics of apomixis, to transfer apomictic genes into hybrid varieties. So certain apomictic genes are identified among plants where they can undergo apomixis naturally without any induction, but induced apomixis also can be carried out, is carried out in hybridization techniques. There is another condition called polyembryony. Normally, a single embryo is present in each seed, but sometimes in certain seeds more than one embryo is present. So this condition is called polyembryony. How can this happen? See, in nature, there are many plants whose seeds have more than one embryo. But in most cases, only one embryo will develop, that is, a zygotic embryo will develop, and all the other embryos usually degenerate. So this polyembryony happens when some of the nucellar cells outer to the embryo sac divides and it protrudes into the embryo sac. So each mass of nucellar cell that protrudes into the embryo sac start acting as an embryo. In certain cases, nucellar embryo would be active. It can grow into a new individual, and in those cases, nucellar embryos are highly significant in horticulture and plant breeding because they are free from diseases. So by that, we have come to an end to the chapter Sexual Reproduction in Flowering Plants. In the following videos, we'll discuss on more objective and multiple choice questions which will help you in NEET. So see you soon in the next video.